and data. Assemble Plus is a European Union-funded research and innovation programme with a consortium of over 20 partners that integrates key marine research facilities across Europe and beyond, offering access to top-tier research infrastructure through a competitive application procedure, evaluated on the basis of a feasibility assessment and research excellence. Whether from academia, industry or policy, through its easy and straightforward application process, Assemble Plus provides scientists with on-site or remote access to biological resources, varied ecosystems, experimental facilities, technology platforms, e-infrastructure and expertise, and provides lodging and catering support over the course of their placement. Assemble Plus also performs its own networking and research activities, ranging from interacting with new users and businesses to cryobanking marine organisms to providing diving services for researchers. Over the course of the project, as well as providing access, Assemble Plus aims to strengthen transnational and multidisciplinary networks, create public-private partnerships, enable new technologies and services, upskill researchers, and improve the long-term sustainability of Europe's marine biological stations. So, if you're a researcher in need of access to marine infrastructure, such as laboratories, equipment, or any other provision, Assemble Plus welcomes proposals for access on a rolling basis from the 29th of August 2018 to the 30th of October 2020. For more information about the project and call for access, please visit www.assembleplus.eu.
Good afternoon, everyone. Welcome back to the Assemble Plus conference 2021. So we are now going to have a series of lectures by visitors of Assemble. So the first one is by Belén González Gaya. She is a postdoc at the University of the Basque Country, working with xenobiotics in the aquatic environment. And she visited CIMAR in Porto, in the north of Portugal. And CIMAR is a member of MBRC Portugal. And the title of the talk is Hospital Waters, Health or Environment? A successful story between chemists and biologists. Please go ahead, Belén.
Thanks for the instructions. I'm trying to share my screen now. I hope you can see it. Let me check.
You just have to click, that's it.
Okay. You see it properly?
It's fine.
Okay, I hope so. Okay, so thanks, Adelino, for the introduction. That was what I was just going to say. I am a postdoc researcher in the Marine Station of Plencia. It's the Assemble Plus node from the University of Basque Country, north of Spain. And I was very lucky to share with my colleague, the PhD student Naroa, 1 month stage at CIMAR in Porto. So for us, it was a great opportunity because as Adelino just said, we come from the analytical chemistry department of our university, and we were sharing our knowledge and learning a lot from a biology group at CIMAR and trying to put together all this knowledge thanks to Assemble Plus. So in this story, what I want to talk about is about pharmaceutical products, disinfectants and other chemicals related with hospitallary origin and the effects that they may pose in the environment. So as you all may know, the analysis of myriads of xenobiotics in the environment stands for a high complex challenge for us, for scientists, because we really want to know, we want to identify them, to prioritize them towards doing a good risk assessment of our environment. In particular, aquatic ecosystems are of great concern as they are subject to the direct release of pollutants from industrial, agricultural, or also urban wastewaters. In particular, hospital effluents are one of the main sources of emerging compounds to the environment because they deliver tons of substances like pharmaceutical products, personal care products, that conventional wastewater treatment plants are not able to remove completely. Thus, aquatic organisms are in constant contact with these cocktails of chemicals that have been found to be toxic, bioaccumulative, but also in many cases, we don't really know. They have non-adverse effects that we still need to study. Then under the umbrella of the One Health Initiative, which stands for healthy people, biota, and environments, this situation needs to be assessed from a holistic point of view. From the chemical analysis point of view, normally we perform targeted analysis. That means that emerging compounds are analyzed, let's say, with directed methods. So we decide what we want to measure and we go and try to find them out. But fortunately, there is an increase on new techniques, non-targeted analysis and suspect analysis that avoids the loss of information because there is no selection, a prior selection of the compounds to analyze. So even if newer and more difficult maybe techniques are needed, the results are promising because these analyses are not biased. Another problem is that normally when we do this kind of chemical analysis is that we forget about the effects or the biological point of view. We only go out to the field, we measure some pollutants, but we don't really care, or we cannot care because we don't know how to, if they are really dangerous, how is the behavior in the environment, are they causing effects on the biota, for instance. So we need some tools that combine these two perspectives. For instance, effect-directed analysis, or EDA, has arisen as a very helpful new tool. Normally, it involves in the same workflow the use of bioanalytical tools. So we use bioassays to assess the potentially toxic effects of a sample, for instance, in our case, of a hospital effluent water sample. We can perform some biotesting on the sample, and then we can do fractionation. So thanks to chromatographic tools, we separate the content or the possible contaminants inside of the sample into different subsamples or fractions, and then we can perform more biotesting again and again. This is an iterative process. So we can kind of isolate the important or the relevant toxicants into few fractions. So then we can analyze only these important toxicants and then go for the identification and quantification of those really causing effects on organisms. For that, within this context, the main objective of our work was the development of an EDA protocol in order to identify the most relevant toxic compounds in hospital effluents, combining different bioassays. Because one of the main achievements of EDA is that we can put together many endpoints at the same time using different bioassays. We will see that at the end of the talk. First, we sampled in the Cruces Hospital, is the biggest hospital here in the Basque Country. For that, we took out of their pipes, so non-treated wastewater, just directly out of the hospital, with a large volume sampler coupled to a solid phase extraction cartridge. This cartridge was filled with different sorbents, which allowed us to gather, pre-concentrate and retain a wide broad of different compounds with different polarities. Then once in the lab, we just have to elute these sorbents, and we got an extract, a pre-concentrated extract with all the different compounds retained during the sample. The fractionation step is also really chemical tool. Is done with preparative liquid chromatography, and we have optimized two different fractionation steps. The first fractionation is done onto a C18 column, and the different compounds retained are just separated along a polarity gradient. In case, well, they are separated in 18 fractions. So the original raw sample from the hospital is divided into 18 small subsamples. In case any of these 18 subsamples is tested and gives a positive response to any toxicity test, we have a second fractionation step on an aminopropyl column. So we have another kind of separation, so we can further isolate the toxic compounds, okay? Just before the bioassays, we need to change the solvent again into water. So there is another preconcentration step onto solid phase extraction, but this is just to maintain the toxicants in water and be able to do the bioassays. Normally we test two different in vivo tests, sea urchin embryo test and zebrafish embryo test. Probably many of you know these techniques, so I will not get deeper. Just the endpoints are the length of the larvae, for instance, or the malformations, or the lethality, the mortality of the embryos when exposed. And in the last year, we have developed also a new protocol using, yes, GS estrogen screening. So we look specifically for estrogenic activity of compounds in the raw waters. So we use a kit with yeast cultures. We grow this yeast, and then we can measure if there is any estrogenic activity. But the thing is, well, first, of course, we need to test if the raw sample is toxic or have any effect, doing a effect dose curve. Of course, you all know what is this. And then we test individually the fractions. The thing is that we are chemists, so we don't really have the know-how of going further on these bioassays. With an embryo test, it's kind of easy because we can observe if they die or not, if they grow or not, but going for a deeper information, more specific, is very difficult for us. So that's why we thought about asking for an assemble research exchange. So we had two previous experiences that were very good. So we decided to look for some place in some nodes where we could do different bioassays, and happily we found one very close by here in Porto with Miguel Santos' team in his endocrine disruptors and emerging contaminants group. So they were doing things very, very similar to us, but from a biological point of view, which was very, very good for us, actually. So let me now go into some biological issues. Sorry if I'm making a little mistake, but this is not my expertise, of course. So what these very smart Portuguese scientists do is to use two kind of nuclear receptors, the ERAX and the PPAR, and gamma the receptors, which are related to the transcription of adipocytes, lipid metabolisms, and some other issues related with obesogenicity. So we could test with their transactivation assays if we had obesogenic compounds inside of our hospital waters. For that, they designed a DN binding domain together with a ligand binding domain with any of the two nuclear receptors. They put it inside of a first plasmid altogether, which will, once it's inoculated into the cell cultures, we have a chimeric nuclear receptor, and we have an internal control with renilla, just a fluorescent marker. If this first plasmid interacts with a second plasmid, then we may have a positive or a negative activation. So for instance, if we have a substance that fits with these transcription nuclear receptors, then the luciferase activates. But if there's nothing that activates it, we don't have a positive response, so we won't be able to measure luciferase. but we will have always the internal vanilla control. So we will know that our plasmids are working even if there's no obesogenic response in the cell lines. In the end, what seemed like magic for us, it was like these nuclear receptors are obtained from humans. They are produced and cloned with bacteria cultures, and subsequently they are transfected together with all the p-bind components and all these small components into a mammalian cell line, with particularly this one in the green African monkey, which is then exposed to the hospital water fractions. So really, biologists, you're amazing. Like for us, this was like a super cool thing to do. We learned to do the seeding, so how to grow cells, how to keep these aseptic conditions in order to grow them. We learned how to transfect them, so how to inoculate the nuclear receptors and all the p-binds inside of the cells. Then we were exposing the cells to our samples, to our fractions. And finally, we were measuring the lucifers in order to see if there was a positive response or any obesogenic compound inside of the fractions. Once back into our lab, into Basque Country, what we wanted to do is to analyze which compounds were in those fractions. For that, we would use high resolution spectrometry with a Q-orbitrap, which allows us to do suspect screening. So we can look for different compounds. Actually, we were using a database with more than 40,000 compounds from the Norman network. But also we can do quantification of the confirmed compounds with around 300 standards, chemical standards that we have in our lab. So we do both suspect screening and target analysis. I will show you briefly, really briefly now, some results of the 4 applied bioassays of the different fractions. In here we can see the growth response of the sea urchin larvae exposed to the 18 fractions. We can see that the raw sample as well as fractions 6 and 7 were significantly decreasing the growth of the larvae. Also this neighboring fraction number 8. Going to the results of the zebrafish embryo test, we can see that also fraction 5, so in the same range of polarity, the compounds in the same range of polarity of those causing effects to the sea urchin embryos are affecting also the fish. So between fractions 5, 6, 7, this is affecting, having a like a non-specific toxic response on these organisms. But then when looking for estrogenicity in the JS bioassay, we see very different results. In this case, fraction 12 is the one exhibiting a higher estrogenic potential. So the compounds retained in this fraction are heavily affecting the estrogenic potential, well, having an estrogenic potential as shown here. Also fraction 6, a little bit, but not really significant. The results for the transactivation assays, in here we see the results in blue for one of the nuclear receptors and in orange for the second one. We see that for fraction 6, so the same as that affecting the embryos of the in vivo organisms, are showing a p-fold value important. We cannot confirm that it is significant by the moment, because when we were in Porto, we got blocked because of the COVID-19. So after 18 days of work, we have to stop with our cultures. So we need still more replicates in order to confirm these results. But hopefully we can go in a close future when everything turns back to normality, and we can really check if there are some obesogenic compounds in these fractions. In the meantime, during autumn and winter, we have been doing the screening analysis. So we know now that, well, we have tentatively identified more than, well, like 30 compounds in the sample, attending to different identification levels according to Szymanski gradient levels. this is something very chemical. What it just matters is that most of them, like more than half, were pharmaceutical products, which turns out to be logical because we are dealing with hospital waters, of course, but also pesticides and other compounds, mainly plasticizers or personal care products, are playing a role into these active fractions. Those quantified with the target analysis at the highest concentrations are antibiotics and anti-inflammatories, but also we cannot neglect, or as I said before, other compounds such as plasticizers that were also detected. By the moment, we have only the results for the first fractioning, so we have still too many compounds in order to blame these effects. So we still need to do the second fractioning. We have already fractionated the active fractions number 6 and number 12. So, and they have been tested for the YES bioassay. So that's why I mean that we have found very interesting results regarding plasticizers. For instance, bisphenol A is present at very high concentrations in fraction 12, and it's affecting definitely the estrogenic activity on there. But also many other of the pharmaceutical compounds are affecting the embryo's growth and malformations. So we still need to do a little bit more to blame some of these guys for all these effects on aquatic organisms. In the meantime, I hope that Assemble Plus carries on and allows people like us to learn new techniques and to share new discoveries with other European partners. So just reminds me to thank all the great team in CIMAR with Miguel Santos, Raquel, and Nesh, which helps us a lot, and the Assemble Plus for funding our trip. And thanks all of you for just listening, and I hope that you liked this talk. If you have any questions, don't hesitate to contact us. And thanks for the invitation of the talk.
Thank you, Belen. So, is there any questions? While there are no questions, I'll ask you a question. So I think it's more than one question, I think, in the end. Do you have any idea of the persistence of these compounds that you tentatively identified in the hospital outflows? So, and the other part of the story is, or the question is, you know, those assays that you are testing, that you are developing, well, that you are using, what you're measuring is essentially acute responses. But, you know, many of these may be persistent, and so the impact goes much beyond sometimes that you can actually see using those methods. So do you have some idea about eventually, you know, looking into the long term?
Yeah, well, regarding the first question about persistency, most of pharmaceutical products, we have to think that they are very active compounds, so they are quite reactive in the media. For instance, now we hear a lot about antibiotics, but it is true that the lifespan of antibiotics in the water is quite low. For some antibiotics, it's just few hours. Well, for others in sediment, for instance, can be even months. But these new emerging contaminants are not just we have to change our mind, because we are not talking about PUPs, for instance. So the problem with these emerging contaminants is that is more related with pseudo persistency. So it's not that they are really recalcitrant, but that they are constantly released. So we have them always in the outflows of our pipes, of our wastewater treatment plants, on our rivers, on our coasts, because we use them at very high concentrations and every day. So the profile of use is more related with their occurrence than the molecule persistency. But still is a field to study, because, for instance, other compounds such as plasticizers, those are more persistent, and we still need to evaluate their lifespan in the environment. Also depending on climate or weather conditions. For instance, in polar areas, the degradation, of course, will be different than in tropical waters. So we need to evaluate that really in the field. And regarding the second question, of course, those are we are not doing chronic exposure, but of course, this is really, really important also to assess. Maybe with this acute exposure, we are going for a first step in the scenario, but of course, cocktails of compounds or chronic exposure is really, really important as well. I totally agree.
Thank you. Nancy Fokadik, you have a question, please?
Yeah, it's a very practical question. I'm a biologist and not a toxicologist nor a chemist. I was just wondering about how much volume you are speaking. So you're sampling, I suppose, only once your hospital effluents, and then you keep the water for the whole set of tests and fractionation. How much water you are speaking about? How much did you take to Porto from Basque Bay?
So talking from a chemistry point of view, then what matters is not the volume, but the concentration.
Right.
So what we do is to pre-concentrate a lot the sample. So with the large volume sampler, what we do is we take samples between 50, 60 liters, integrated samples, because we can program like cycles of the sampler. So during 24 hours, we can sample maybe every half hour or every hour just for 20 minutes. So we have a full 24 hours sample, integrated sample. And then what we do is just to pre-concentrate it a lot. So normally the enrichment factor of our samples are like 1,000 times the original one. So to Porto, we took a small box of vials with 1,000 times concentrated fractions. And then we do this dose effect response, testing different concentrations. So we find the EC50s or LC50s, EC75, and then we decide at which concentration we can see some effects. And then we test with that concentration the different fractions.
Thank you.
Sorry, I have a little technical hitch here. I'm going to try to correct it. Sorry, I have to leave for a second because otherwise you'll be thrown out of the meeting.
Okay, so as we don't have any more questions, no? Okay. So perhaps we can try to prepare everything for the next session. That will be José Martin Duran. José, are you there?
Here.
I think, I'm not sure if it's okay or not, but I think now it's going to survive. So is there any other question?
No.
No. Okay, so we'll move on to the next speaker, and it is my pleasure to introduce José Martin Duran, a lecturer at Queen Mary University in London, and he is interested in the evolution of animals with special focus on invertebrate diversity, major evolution transitions, in particular the origin of bilaterian animals. And in a sample, he visited the marine station in Roscoff, and today he's going to talk about establishing the annelid Owenia fusiformis as a reference species for evolutionary genomics and evo-devo. Please, José. The sound seems to be off. You should be able to control where the microphone is. Choose the speaker, the microphone. I can hear you now.
Hi, do you hear me?
I can hear you.
Because it's which is breaking. Sorry. Well, thank you for the invitation. Yeah. So I will start sharing my screen. I can, share. Okay, so I guess now you're seeing the full screen. Okay, well, as I was saying, thanks for the invitation and thank you for the introduction, Adelino. It's a pleasure to be here and contributing to this meeting on Assemble Plus and explaining some of the experiments and investigations that we've been doing with the help and support that we've got through the years via the Assemble first and then the Assemble Plus program. So we are biologists, we are not chemists. We work on actually embryos that the name was showing in the previous talk. And we are interested in understanding the evolution of animal diversity. So my lab is interested in understanding why animals are diverse, why we see morphological diversity. And the way we address this question and we try to investigate this fundamental biological question is by comparing the embryogenesis of different animals, by comparing how animals develop and the different stages. So here you have a snail and a rat or a mouse. And then by comparing how the embryos of these animals develop and finding similarities and differences, we might be able to understand how these two different organisms, how these two different animals evolve and understand the different processes that occurred during time that led to the evolution of these two animal lineages. And in particular, we are interested in understanding the evolution of annelids, of marine invertebrates, and in particular, annelids, segmented worms. And this is a tree showing the main lineages of segmented worms, and just with 3 drawings showing that segmented worms can be very, very different in terms of morphology. If you want to just picture one, think of an earthworm. But the way we try to understand and approach this question is by studying this particular annelid that is Owinia fusiformis. So Owinia fusiformis is key to understand animal annelid evolution and also generally animal evolution because it's a member of the Paleoannelida. So it's a member of the sister lineage to all remaining annelids. And therefore, by comparing Owinia fusiformis with other deeply nested lineages of annelids, of segmented worms, we will be able to infer common and ancestral traits to Annelida. And therefore, we will be able to trace the evolutionary pathways that key morphological and developmental features follow during evolution of segmented worms. And we, one of the main reasons that we took, one of the main reasons underlying the decision to study Owinia fusiformis is that well, and this is just to show that actually it is a Paleoannelida because it's thought to actually represent the most ancestral annelids. So as I was saying, one of the reasons to pick Owinia fusiformis as research species is because it was available through Assemble and Assemble Plus, because it's easy to collect, it is easy to find and it's easy to collect through the network of marine stations. And is available as a wild research species via the Station Biologique de Roscoff. So we, since 2014, we've been regularly accessing the services provided by Assemble Plus and collecting mostly through remote access hundreds of animals, hundreds of Owinia fusiformis during the spawning season, during the breeding season of the animal that happens in spring and summer. And that has allowed us to establish as always the same population, a single population of Owinia fusiformis as the world reference for evolutionary and developmental studies. And definitely this would have been way more difficult to do without the support and the facilities offered by this program, by Assemble Plus. So what is exactly what we are trying to understand? Well, in particular, we are trying to understand how annelid worms, segmented worms, and there you have an earthworm, develops and evolved. And segmented worms belong to a very large clade of animals comprising 16 different animal phyla, so almost half of the animal kingdom, that is called the Spiralia. And within the Spiralia, we have different key animal groups, like for instance, flatworms and snails, mollusks, but all share, or at least in the ancestral, as an ancestral feature, the presence of a common developmental mechanism, a common developmental strategy that is called spiral cleavage, and hence the name Spiralia. So it is very interesting, and it's quite fascinating, at least for me, to try to understand how, from a very similar development that is called spiral cleavage, it has evolved an extreme diversity of animal lineages, from segmented worms to flatworms to a snail inhabiting the terrestrial environments. And this is the major broad question that we are trying to address by studying Owinia fusiformis that we get through the assemble. In particular, we are interested in understanding how the spiral cleavage works, is controlled at the molecular level and at the genomic level. And specifically, we are interested in understanding how the main founder lineages of the different tissues of the animals are established at the genetic level, and how these different and how the mechanism controlling this developmental process evolved. So, being very simplifying things, because I guess the background of the audience is quite broad and maybe not even related to biology. These organisms, mollusks, annelids, they specify 4 major founder lineages after the first 2 rounds of cell division, after the zygote is formed, after fertilization. These founder lineages are named A, B, C, D, and each of them, the progeny of each of them will generate roughly a quarter of the animal. So, for instance, the D lineage will form the posterodorsal side of the animal, the A will form the left, the C will form the right, and the B the anterior and ventral. And there are, although all these different animals follow the same kind of development, this spiral cleavage, the way they specify these 4 different founder lineages is different. In some cases, they follow a conditional mode of specification, and in some species, they follow an autonomous. In the conditional way, the specification of these 4 actually happens quite late in development through interaction between cells. Okay, it's kind of a self-organized mode of specification, and therefore, the beginning of development, the first 4 cells, the cells are not specified, so they are totipotent. Okay. However, in the autonomous, the mother controls this process and speeds it up. So the mother loads maternal determinants in the oocyte that then get segregated differentially between these 2 first zygotic divisions, and this differential segregation is what drives the specification of the 4 A, B, C, D lineages. Okay, so we are interested in understanding these using Owinia fusiformis. And why Owinia? Well, Owinia is important because it shows conditional spiral cleavage. Okay, here you have this is a 4-stage cell embryo for Owinia, 4 and 8, and you see that all cells are equal size, and actually all cells are totipotent. They are equal. They have the equal capacity to develop the animal. We've been characterizing the development of Owinia, and we know how embryogenesis happens and takes place, all the different stages. So first, the embryo will form this ball of cells, and then the ball of cells will form the main different tissues, and then there will be a phase of organogenesis and a development of a larva, planktotrophic larva, that will swim in the water column, eat plankton mostly, and then at some point grow and decide to settle and metamorphose into the adult. Okay. So now we can put all things together and get basically this phylogenetic comparative framework that we are using in my lab to try to understand spiral cleavage evolution, and in particular the evolution of segmented worms and the development of these animals. So Winia fusiformis, as said before, is the sister lineage to all to the remaining annelids. And here you have 3 different annelid species that are commonly used in Ivodivo. They all have the autonomous cleavage, and therefore using Winia, that is the sister to all of them, with conditional equal cleavage is important as to use it as outgroup and therefore define ancestral and derived characters for segmented worms. So that's the principle. Then the question that we are trying to understand, my lab is trying to understand, is how conditional spiral cleavage is controlled and organized in Winia. So we started focusing on MAPK signaling, and this is actually one of the projects that we proposed to do through Ensemble Plus. And it was focusing on MAPK signaling pathway. So it doesn't matter the name, but it's simply a signaling pathway. So a way cells have to communicate that instructs specifically in mollusks the cell, a particular single cell, to become the D lineage. And this is well conserved between different mollusk lineages. And it was shown a while ago, so almost 20 years ago, that at least an annelid species also seems to use this signaling pathway to specify, the D quadrant. So of course, here came the question, is Winia as the sister lineage to the remaining annelids also using the MAPK signaling pathway, this signaling pathway, to specify the D quadrant, so one of the founder cells. So we collected embryos, we collected animals through an Ensemble Plus, then we used them to obtain embryos, and then to track during developmental time the localization of active MAPK, so the activity of this signaling pathway, and we found here you have it with
Shema, it stopped. It stopped.
We can see that actually it is very localized during early development, during the 32 cell stage to the late blastula, and at a particular stage of development that is 5 hours after fertilization, similar to other mollusks, MAPK, the signaling pathway, becomes active in one single cell, in one single cell. And then here, this would be the summary, and you can see that it's actually very similar. The dynamics of activation of this MAPK, of this signaling pathway, is very similar to what has been described in mollusks, further supporting this idea that the spiral cleavage is actually a very common and conserved developmental process for spiralians. Of course, what we wanted to test is, okay, now we see that actually this pathway is active in Winia, but that is this pathway controlling any developmental process. So we can actually target this signaling pathway by using chemical compounds. So we can either use this BFA chemical compound that will block cell-cell communication, or we can target directly the pathway using a drug that is named U0126 that will block the activation of MAPK. And what we when we treat embryos with these 2 drugs, we see that actually we lose completely the activation of MAPK, which is further supporting that actually our treatment is successful and efficacy. Okay, but what happens to the embryos when we block the activation of the MAPK signaling pathway? Well, actually, what happens is that they become radialized, so they are unable to generate posterior structures. Okay, so here you have the control on the left, and then you see that the control is elongated from the mouth to the anus, and then it has a kind of a tail with kitty, like that would be like long needle-like structures that could be seen as defense. When we treat with these two drugs, when we block the MAPK, they are unable to develop the KT and to elongate along the anterior posterior axis, and therefore the embryos become completely radialized, just simply showing a single opening that is the mouth, as you can see here, confirmed by the expression of an oral gene or a marker, a gene that is expressed around the mouth. We can see here, perhaps better through morphological analysis, how the larva just becomes a hat shape, only having a central opening that is the mouth, and then still keeping the ciliated one. So we follow up. That's basically the treatment that I show you. But we followed up and try to understand more about the dynamics of the MAPK signaling pathway and their role during early development. And we did some nested shorter windows of treatment that allowed us to actually identify 2 periods of development where MAPK is important, and actually having 2 different roles in the specification of different structures and tissues. An early role during 2 to 3 hours post-fertilization, where actually the pathway is probably required to specify the apical organ, so the kind of a most anterior tip of the embryo, and then a later one where this signaling pathway is essential to specify this D quadrant that will be used as a signaling organizing center to pattern the entire embryo later on. So basically, this was key to propose a scenario regarding the evolution of the control of the spiral cleavage in Analytes. So we now are able to show, we can now conclude that Owenia fusiformis indeed shows the activity in a single cell controlling the organization of the embryo during early development, as it also found in mollusks. And therefore, probably this is telling us that that was an ancestral character for Analytes. And most likely also the presence of these properties in Analytes and mollusks is also suggesting that it's an ancestral, considering the topology of the Spiralia, that this is also an ancestral character for the entire animal group. We also then wanted to take this study one step further, and then trying to understand what are the genes and the gene regulatory networks that are beneath that are downstream of the MAPK signaling pathway, and that are actually controlling the role that this signaling pathway has in Analyte development. And to do so, we applied high throughput next generation sequencing. So we repeated the same drug treatments to block the MAPK signaling pathway, but then collected samples to do an unbiased high throughput screening of the genes that are expressed at 2 time points in control and treated embryos. The first time point that we sampled was right after the activation in one single cell of the MAPK signaling pathway, 5.5 hours post-fertilization. And then the last time point, the second and last time point, was in the larva, where we could see the morphological effect of the treatment. Once you have the next generation, once you have this high throughput data, you can map it to the gene repertoire of the animal and then infer genes that are differentially expressed between control and treated conditions. And I will just simply go quickly through this data. But interestingly, we observed the presence of 2 genes that were common to all different treatments, and therefore that we deemed them as key and crucial to control the phenotypes that we see when we block the MAPK signaling pathway. One of these key genes was caudal, CDX, that is an evolutionary conserved gene controlling the development of the posterior structure. So our tails, during our development, our tails are express caudal. And then we could show that actually this gene also in Owinia is expressed in the tail hindgut region of the larva and the adult. So that was actually good, that was positive, because it reassured us that the genetic mechanisms that control some of the development of some of the structures that we lose in Owinia after treatment are similar to the ones that we see in the development of equivalent structures in other organisms. We also found that twist, again, another evolutionary conserved gene in the development of muscles, and mesoderm was also affected by treatment with drugs against the MAPK signaling pathway, which is actually supporting a situation in which MAPK is activating the cell that controls the organizer, and then the organizer cell, the D lineage, is driving the development of the hindgut and the trunk mesoderm. Interestingly, this is just to show how the activation of the D quadrant, the activation of the MAPK signaling pathway is key to actually activate a vast cascade of downstream genes. So you see here the line, the blue line is marking the 5 hours, just right after 5 hours. And we see how many of the genes that we find downstream in our RNA-seq comparison between treated and control are actually peaking in their expression, so high expression is in red, are peaking in their expression right after 5 hours, further suggesting that they are actual downstream targets of MAPK. And interestingly, above all the different genes that we found affected, there was one that was heavily differentially downregulated when we were blocking MAPK signaling pathway, and this gene is called FOXH. And what is interesting is that FOXH is well characterized in us, in our invertebrates, humans and fish and xenopus, but it was long thought to be actually absent from other invertebrates until some genome analysis found it in a couple of mollusks, Lotia, limpet, and then the oyster. But actually the role of this gene during development in invertebrates is unknown. And it was actually thought to be absent from other annelids like Capitella and Helopdella. So it was actually a very, very surprise finding to find FOXH, this gene that was thought to be absent in many invertebrates, severely affected by the treatment of MAPK. We could identify the dynamics of expression of FOXH, and then you see that actually it peaks again right after 5 hours, further supporting that is controlled by MAPK. And then we could do the in situ hybridization, and we could check the expression of FOXH. And we found that it was initially expressed in just one cell in the blastula at 5 hours. And then it was expressed in a very localized 2 pairs of cells during gastrulation, and then completely off in the larva. Again, all supporting the fact that this could be a direct downstream target of MAPK signaling pathway involved in this very early role of patterning the embryo. And this is actually very, very similar to the role that we know for FOXH in invertebrate development in zebrafish, for instance, where FOXH is expressed in the organizing node, and in particular in the developing mesoderm. Okay, and well, at the moment we are trying to expand and investigate further on this particular finding. So I think it's almost 25 minutes. So just conclusions, the take-home messages. Well, the constant support assemble and assemble class allow us to establish Owinia fusiformis as a research system to study equal cleaving in annelids, and especially providing the community with resources of a slow evolving genome and a slow evolving annelid. We found that a MAPK signaling pathway controls the development of Owinia fusiformis and specifies the organizer. And this is quite similar to what we also know with mollusks, and therefore is probably an ancestral character for all spiral cleaving organisms. And we have also identified that MAPK exerts this organizing activity by triggering a conserved posterior mesodermal program that is also similar in some aspects to what we see in other animals, like for instance, us, vertebrates. And with this, just to say thank you all for your attention. Most of the work that I presented was done by these three people in my lab, mostly Ocean and Alan, and it's supported by, of course, Assemble, but also my institution, Queen Mary, and a starting grant from the European Research Council. Thank you, and happy to take any question.
Thank you, Shema. Very interesting. Is anyone wants to ask a question? I have a bit of a peripheral question because I don't really know much about the subject. Is how important is epigenetics in terms of development of these kind of organisms?
So yeah, that's actually a very, very important and interesting question. And we are looking deeper into the epigenetics aspect. It is for sure critical, and one of the hypotheses that we are trying to tackle is whether epigenetics is one of the main drivers in explaining the differences between conditional and autonomous. Whether this autonomous mode of developing that is kind of, controlled by the mother, and then it's just, okay, I don't need to communicate, is controlled by epigenetic factors that the mother loads. And therefore, these epigenetic factors are already triggering and controlling everything. While in the case of Owinia or conditional annelids, the role of epigenetics happens later through cell-cell communication and through a self-organizing process. But yeah, definitely.
Thank you. So we've got two questions here. Elizabeth, can you ask the question?
Yes, sure. Hi, Gemma.
Hey, Elise.
I was just wondering what happens to the MAPK-treated Owinia later. I don't know if they have feeding larvae, but do they feed at all? And do they have a normal-looking apical organ structure?
Yeah, that's good. Good question. So I think they don't. I don't think they feed. We haven't tried to feed them, to be honest, because it does have midgut, so they do keep. We have checked the expression of genes like GATA, so they have a stomach, a midgut, but we don't know if it's functional, and yeah, I doubt it. And they do have some signs of apical organ, but it depends on the window of treatment. So if you treat from 0 to 5, you are also blocking this narrow window of time before the specification of the quadrant, which it is required for apical organ specification. So depends on when you treat the larva with the radialized larva will have apical organ or not, or at least signs of an apical organ. Yeah.
Okay.
Ulf,
thanks.
Ulf, please go ahead.
Yes, thank you. Well, first of all, thanks, Gemma, for an excellent talk. Really interesting. Hi.
Thank you. Hi.
I just have a sort of a general question, which is sort of very basic. But I mean, if you look at this Owinia, to me, it kind of looks like any other polychaete, like maybe Maldanide or something like that.
Mm-hmm.
So is there anything in the general morphology like that sets all the other annelids apart? Is there any synapomorphism that you could point to that indicates that this is the group?
That is, you mean like morphological synapomorphy that supports the phylogenetic position?
Yeah, exactly. Yeah.
Okay. So I think this. I think it was surprising to find out that the Owinids and Magellonids were the sister lineage to the remaining annelids.
Yeah.
I think. I'm not a morphologist, so I don't know whether there is actually one synapomorphy in terms of morphology that would support that. However, I think that in terms of molecules, it is really, really, really consistent. Yeah. We are exploring that in terms of genomes. So try to see whether there are, for instance, rare genomic changes. that would support that by comparing with outgroups, like for instance, my mollusk. And we found some hints that perhaps would show that there are things that you would expect in the ancestor to annelids still retain in Owenids and lost in others. But yeah, in terms of morphology, I mean, most likely you or any others know more in terms of morphological features that might support that position.
Okay, thanks again for a great talk.
Thank you.
Thank you very much. Well, I don't see any more questions, so we move on to the next speaker. Thank you very much, Shema. Yep. That was very good. And now we're going to listen to Jan Rubens. He is a postdoc at the Flanders Marine Institute. And this is not result of a sample visit. I just thought that it would be interesting to bring a slightly different subject from what we normally have. And also, of course, it's also provides an important view of research infrastructures, in this case, to track animals. So, in particularly fish. So, Jan, please go ahead.
Thanks, Adelina. Thanks for the introduction. So I'll share my screen. Okay, so I should be sharing now. So welcome everybody, and thanks for joining, and thanks to Assemble for giving me the opportunity to present here at this conference. It's my first online conference. And I'll start by, by showing you this picture. What you see here is not the illumination at night, but it's a heat map. And it's a heat map showing where in Belgium and in the Netherlands people are running. And if you zoom in to the map, this is Ghent, the city where I'm living. And the red dots is where my house is. And then you can see that there's many people that go running along this track. And this place it's a river. And it's a place where I also go running myself, and it's a beautiful place for running. So this is just an example to show you that based on information of tracking people, you can get interesting insights of what people are doing and why they are moving, and how this is linked to your environment. And this is all linked, of course, based on this GPS technology. So it's a very interesting technology to know more about behavior. And of course, we don't only use this for human behavior, but also for animal behavior. So using this tracking technology, we can get insights on ecology and movement behavior and how animals are linked to their environment based on tracking them. And there's a multitude of species all across the world that are being tracked. And on land, we mainly use this GPS technology as this is the most easy technology to use on land. However, if we go to the aquatic environment, then we have a problem because these GPS signals don't work underwater. So if we want to know more about tracking fish and how they move and why they move, we need other techniques than this GPS technology to be used underwater. And there's a multitude of techniques that can be used. And in this figure, I show the 5 most used techniques, which are radio telemetry, PID technology, satellite tags, acoustic telemetry, and archival tags. So we use these 5 types of technologies to track animals underwater. In radio telemetry, we use radio waves, and then we have antennas to listen to these waves. In acoustic telemetry, we use acoustic signals that are detected by receivers. In PID tags, it's a bit the same as an acoustic telemetry, but there the tags are activated by an electromagnetic field, and then in satellite and archival tags, we actually store the data on the tag. So you have sensors like depth sensors or temperature or illumination that are measured on a fixed delay, and then all this information is stored on the tags. And then in satellite, this information is sent to a satellite to get your data when the animal surfaces. And in normal archival tags, you have to retrieve the animal or the tag manually to get to your data. And in all of these techniques, there's one thing in common, and that is that you always need to attach or implant a tag to an animal. Of course, this might all sound a bit difficult to understand, so that's why I have a short movie to explain in some more detail how these technologies work. The movie is focusing on acoustic telemetry, but it can be extended to the other technologies as well. So just watch for a minute.
Aquatic ecosystems are full of life. Atlantic salmon migrate in and out of rivers to spawn. Sharks can move long distances in the ocean. Lobsters move around the ocean floor foraging. Eels migrate incredible distances to spawn at sea. Sunfish roam the wide ocean. Tunas can dive down to more than 1,000 meters. Understanding where, when, and why animals move is necessary to adequately protect and manage them in a sustainable way. On land, we can use GPS to track animals, but that doesn't work in water. So how can scientists get this information when animals live underwater? One way to do that is using acoustic telemetry. Telemetry means to measure from a distance, and acoustic means using sounds. So scientists use acoustic telemetry to track animals from a distance using sounds. Once the study animals are caught, they undergo a small surgery so the scientists can implant acoustic transmitters. For larger fish like sharks and tunas, or for animals with a hard shell like lobsters, the acoustic transmitters are attached externally. These transmitters emit a sound through a sequence of pulses. The sequence is unique for each tag, so each animal has a unique ID. The IDs can be detected when the tagged animals swim close to receivers, as the receivers detect the sound emitted by the tag. Researchers have a network of thousands of receivers across Europe to track aquatic animals. The receiver information provides data on when and where animals move, but it can also assist scientists in understanding why they move, such as for spawning or feeding, or even figure out where they are seeking refuge. The tagging research can help managers in decision-making processes, like where to create marine protected areas or...
Okay, so I'll stop the movie here. So it's just a nice introduction of one of these technologies, but it's similar in all of them. So you have this tag that needs to be implanted on the animal, and then you have specific devices that listen to the signals that are sent out. So in radio telemetry, you have your antennas. In acoustics, you have the receivers. And in PIT technology, you also have these stations with the electromagnetic fields. And then it's only in this archival tags and in the satellite tags that you just store the information on your device itself, and then you need to get it back. So, okay, this was a short introduction on how the technology works, what you can do underwater. But as a background, it might be interesting to realize that there has been a tremendous evolution in this technology. Already centuries ago, people were also interested in what aquatic animals were doing. Of course, then we didn't have the advanced technologies as we have today. And then people used, for instance, ribbons that they attached to the tail of a fish to investigate whether the fish was coming back to the same location to spawn in the next year. So this is the very early stage of tracking animals. Later on, we moved to these conventional tags, where you just attach an external label to your fish and you get some information on where the fish is captured and recaptured. And then we moved to this electronic tags. And the very first fish being tagged in the North Sea was a place, and it was used, it used sonar. So they had this big boat following one tagged fish for a couple of days. It provided very interesting information, but of course it's very expensive to have a very big boat with a number of crew members on board and just following one fish. So luckily nowadays we have these more advanced technologies to investigate this movement behavior. And it's fast evolving, not only the technologies itself, but also related to the tag size, the battery consumption, and the sensors attached to it. Okay, so now we know a little more about how we track animals underwater. I will go in somewhat more detail of what we do in Belgium. And all the work we do in Belgium is related to this LifeWatch project, LifeWatch Belgium, in which we have this observatorium that we use to, for instance, monitoring this fish movement behavior. And in Belgium, we use two of these tracking technologies. On the one hand side, we use this acoustic telemetry, and on the other hand, we use these data storage tags. If I have a look to this acoustic telemetry, we build out a network in the framework of LifeWatch, where we have a number of stations, a number of receivers where we can detect the tagged fish. And the network extends from the Belgian part of the North Sea towards an estuary and then some rivers on land in Belgium. And it's a network of over 120 receivers. And in the past years, we've been tagging a multitude of species. So here you can see a list of species that has been tagged, and the numbers in the figure indicate the number of individuals per species being tagged in each of the years. And then the color code represents the percentage of the fish that has been detected on the network. So we have this multitude of species, and it gives some very interesting insights. So now I will zoom in some of the results we got from this network. Here on the left figure, we zoom in on the journey of just one sea bass. So the sea bass was tagged in late autumn, close to the shore, and then it went offshore to warmer waters. It was being detected at an offshore wind farm, and then a couple of months later, it popped up at the shipwreck. After a while, it came back to the wind farm before it went back to the shore. And then it went towards the estuary, and then half a year after it has been released, it was recaptured at almost the same location where it was captured. So if you would use this conventional tagging methods and just have this capture-recapture information, you would say, okay, this animal hasn't moved, it just stayed in the same place. However, using these tracking technologies, we see that the animal changes its habitat based on some environmental clues related to its in the lifetime. So this is one example of one tagged fish. Another example is, for instance, that we had eels being tagged on different places in Belgium, the Netherlands, and Germany. But all of the eels moved through the rivers towards the ocean, and they all passed our Belgian network in the same period of time. Within a couple of weeks, all the animals from the different tributaries, from the different rivers in Belgium, the Netherlands, and Germany were seen on our network. And for eel, it was believed that they moved towards the Sargasso Sea, going over the United Kingdom. And, because you have the best, let's say, the best currents for movements going over the UK. So for a long time, everybody was thinking that they use this way to leave the North Sea. But we showed with our network that at least a part of the population goes through the southern North Sea and the English Channel towards the Atlantic. So this is another interesting insight, that based on this tracking information, you get new information on the behavior of your species. Another example is based on plays that we've been tagging in a wind farm to assess whether these animals have any link to these artificial structures at sea. And so on the left-hand side, you see the wind farm, and the black dots are the receivers where we can detect the animals. And then on the right side, you see the presence plots of each of the individuals. So each line is one individual, and you can see that most individuals were present from the beginning of summer until October. And in October, we maintained the receivers. We did an interim data download, so the study is still ongoing, but we can see that most of the fish, at least already until autumn, stayed in the wind farm, and so show a very high residency related to these wind turbines. Now we are waiting to go at sea again and to see whether they moved out in winter time and when they did so. And then the last example linked to this acoustic telemetry is that we can also use it to assess anthropogenic impacts to the animals. Here we have the results of a seismic survey that has been performed at sea, in which we investigated whether or not Atlantic cod reacts to these seismic activities. And we could link movement behavior of the fish based on the data we had. And the model shows that cod can have different behavioral states, being inactive, locally active, of transiting. So inactive is that they don't move lots. Locally active is that they move a little, but they do small distances. And then in transit, you see that they are just going for longer distances. And we've seen that linked to the survey that before there was seismic activity, there was a percentage of inactivity and local activity. And as soon as the exposure started, the percentage of inactivity of the cod increased a lot and the activity decreased. So this is clearly showing that based on this human activity, it influences the movement behavior of the fish. In addition, we could also see that after the survey, the fish were further away from the turbines. So we do know that cod is attracted to the turbines at sea, but after this seismic survey, apparently they moved a bit farther out, and we expect that this is linked that to the behavior, that they start roaming around and are assessing whether or not to leave the area. So again, some interesting results. So that was some examples related to acoustic telemetry and the range of studies that can be done. And now some more insights, some more stories related to this data storage tags we do. This is something we started more recently, so we don't have that much results yet, but we do have some stories worth telling. For instance, this figure here is from a tag that was attached to an eel, and the purple dots represent the depth and the black dots represent the temperature. So you can see the animal was tagged in December 2018, and you see it moves around in the water between sub-zero up to 15-20 meters deep, and you see a slow increase in the temperature. But then all of a sudden you see that temperature increases by more than 10 degrees. And shortly after that event, the depth decreases to 0. So the tag surfaced. So what happened here is that we had a predation event. So this eel has been eaten by a predator. And based on the temperature information we have, we assume that this eel was eaten by a basking shark. And then shortly after being eaten, the shark expelled the tag, and then the tag floated to the surface. So we don't only get insights in the behavior of the study animal, but we can also get some interesting information on the predators related to it. Another story is from a sea bass that was tagged in an estuary. And a couple of months later, it was recaptured in the same estuary, only a couple of kilometers away, I think 1 or 2 kilometers away from the capture site. However, sea bass is expected to leave the area in the winter. So the animal was tagged in October, and we expected it to leave, as normally sea bass wants to go to warmer waters, and so we expect them to go to the Atlantic in winter time. But based on the depth information we have and the temperature information, we were quite sure that the animal stayed in the neighborhood, that it didn't move out. However, if you look to the temperature, we see temperatures between 15 and 20 degrees, which is much warmer than the water we have in wintertime. In wintertime, the water here is about 3 to 7 degrees. So what we suppose that the animal did is that it moved to the cooling waters, the outlet of cooling waters of turbines in nearby the area. So instead of moving out of the area to warmer waters, it stayed in the area and found warm waters, again related to human activities. So which is also a very interesting insight related to management of the species, as we always expect that sea bass is not present in our areas in winter time. So this is just a picture of a data storage tag. And I want to end this talk by just mentioning that, of course, what we do in Belgium is not isolated of what is done in the rest of Europe. And that's why we started creating this European tracking network. We want to connect all researchers tracking aquatic animals in Europe, and we want to unite them in one pan-European collaboration. This European tracking network has been developed a couple of years ago in a project Mission Atlantic, in Project Atlantos, and is now funded under a cost action. And the goal is to bring all researchers together, as fish, of course, don't take into account the national borders we have, and they just go from one country to another. So there's still a lot of data we can gather by just cooperating on these aspects. We have a central database for the European tracking network, and we currently have over 240 users in the system. There are more than 75 institutes involved, and it already has data of over 7,000 animals that are being tracked across Europe. Okay, so this was it. It was just a short trip around Europe, let's say, and to highlight some cases, some study cases we have, and how the work we perform, how it works. I thank you for your attention and can take some questions.
Thank you, John. So any questions to John about this topic, tracking fish? I have a small question. So how small can you now track? You know, how small a fish can you track? Because of course there will be different behaviors according to development, etc.
Yeah, it depends a bit on the technology. So there's different technologies you can use, and also using different frequencies. But nowadays there exist very tiny tags, so you can already tag fish of, I would say, 5 to 10 centimeters. Of course, depending on the technology you use. But it goes from the very small to the very big animals.
Yeah, very nice. I'm curious about the eel stories because we want to know more about eels all the time. Okay, so well, thank you very much. It doesn't seem as if there are any other questions. Our next speaker will be in 5 minutes, so we better wait because there'll be people logging in, I guess, for that purpose. Thank you.
Okay, thanks.